I'm Dr. K.AMUDHA from Biochemistry department. In this topic, we can see the serum marker enzyme in heart disease and the enzyme as a therapeutic agent. So coming on for the serum marker enzyme in heart disease, the major enzyme which is elevated during heart disease are aspartate transaminase, creatine kinase, or else you can say this also as creatine phosphokinase, that is CK or CPK, and the other enzyme is lactate dehydrogenase, that is LDH. The other enzymes are histaminase and cholinesterase. So we'll see the enzymes one by one. Aspartate aminotransferase, that is called as a AST. This enzyme converts aspartate and alpha ketoglutarate for the formation of glutamate and oxaloacetate. And this enzyme is also called as serum glutamate oxaloacetate transaminase, that is SGOT. This AST needs pyridoxal phosphate as a coenzyme. The normal level of AST is 8 to 40 units per liter. So when you take the distribution of this enzyme, this is distributed in liver, heart, skeletal muscle, kidney, and also in erythrocytes. The marked rise of this enzyme is seen during circulatory failure with shock and hypoxia, during myocardial infarction, and also during acute viral or toxic hepatitis. The moderate increase of this enzyme is seen during cirrhosis, cholestatic jaundice, during malignant infiltration of the liver, during skeletal muscle disease, and after trauma or surgery. That is especially after cardiac surgery. The AST level in myocardial infarction. This enzyme during myocardial infarction sharply rises within 12 hours and reaches the peak in 24 hours and returns to normal within 3 to 5 days. The next enzyme is creatine kinase, that is CK, or else creatine phosphokinase as CPK. This enzyme catalyzes the conversion of creatine to creatine phosphate. Normal serum levels of this enzyme is 15 to 100 units per liter for males and 10 to 80 units per liter for females. This CPK consists of 3 isoenzymes. Each isoenzyme of CK is a dimer which is made up of 2 polypeptide chains. The molecular weight of this enzyme is 40 kilodaltons. The subunits are called B for brain and M for muscle. So we'll see the isoenzymes of CK. CK, I have already told, it is a dimer and each subunit molecular weight is 40,000. The subunits are B for brain and M for muscle. Normally, CK2 is only 5% of the total activity. The isoenzyme MB is important in myocardial infarction. The important enzyme in energy metabolism. And this enzyme is an immediate source of ATP in contracting muscle. Here, 3 isoenzymes are separated by electrophoresis. They are CPK1, CPK2, another one is CPK3. CPK1, it is also called as CPKBB. That is, it is found mostly in the brain and lungs. The next is CPK2. It is also called as CPKMB, and this is found mostly in the heart. The last isoenzyme is CPK3 and is also called as CPKMM. This is found mostly in the skeletal muscle. the CPK and heart attack. The isoenzyme CPK2 is very small, that is 2% of the total CPK activity. And so since it is very small and it is undetectable in the plasma, during myocardial infarction, that is during MI, the CPK2 levels are increased within 4 hours, then falls rapidly. The total CPK level is elevated up to 20 folds in MI. We'll next go for the next enzyme, that is lactate dehydrogenase, or else it is LDH. Lactate dehydrogenase reversibly converts lactate to pyruvate in different tissues. This lactate with NAD, it is converted to pyruvate and NADH plus H. And this is done by the enzyme lactate dehydrogenase. The molecular weight of the enzyme lactate dehydrogenase is 32 kilodaltons, and this is a tetramer, that it is made up of 4 polypeptide chains, 2 of M and other 2 of H. So M is a muscle and H is heart. This LDH consists of 5 isoenzymes. They are LDH1, LDH2, LDH3, LDH4, another one is LDH5. These isoenzymes are separated by cellulose acetate electrophoresis at pH 8.6. The normal value of LDH in serum is 100 to 200 units per liter. The values above the upper range are generally seen in children. Now we'll see LDH isoforms. LDH enzyme is a tetramer with 4 subunits. The subunits may be either H, that is heart, or M, that is muscle, polypeptide chain. So when you see the isoenzymes, its composition, its electrophoretic migration, and where it is present and when it is elevated. We'll see one by one. So LDH1, the composition, it consists of fully H chain, that is H4. This moves very fast in an electrophoretic migration, and this is present in myocardium, RBC, and in kidney. This is elevated during myocardial infarction. So coming on for the next isoenzyme, it is LDH2. The composition is H3 and M1. This is present in myocardium, RBC, and in kidney. This is elevated during kidney disease, megaloblastic anemia. So coming on for the next isoenzyme, that is LDH3. The composition of this isoenzyme is H2M2. This is present in brain. This is elevated in leukemia and in malignancy. Coming on for the next isoenzyme, it is LDH4. The composition is H1M3. This is present in lung and spleen. This enzyme, that is this isoenzyme, is elevated during pulmonary infarction. Now coming on for the last isoenzyme, this is LDH5. The composition is M4. This moves very slowly in the electric field. This is present in skeletal muscle and in liver. The elevated level is seen in skeletal muscle and during liver disease. Next we'll see the cardiac markers for diagnosis of myocardial infarction. So there are certain markers in the sense there are certain enzymes which is increased during the myocardial infarction. So we'll see one by one all the enzymes and how it rises, its peak value, and the time how it comes to the normal, and the remarks, etc. So the first cardiac marker, when you see, it is creatine phosphokinase, or else you will be saying this as CPK or CK. This rise after 6 hours and it reaches the peak level at 24 to 30 hours and it returns to normal in 2 to 4 days, usually after 72 hours, and this represents the early marker and cardiac specific. The next enzyme is aspartate transaminase, that is AST, or you will be saying this as SGOT. The early rise is seen within 12 hours and this reaches the peak level at 24 hours and it returns to normal in 3 to 5 days. And the remarks for this is relatively late cardiac specific but can rise in hepatitis. The next important enzyme is lactate dehydrogenase, that is LDH. The early rise is seen within 12 to 24 hours and it reaches the peak value in 2 to 4 days, that is during 48 hours, and it returns to normal in 8 to 14 days. And the remarks is relatively late marker and it is non-specific. So coming on for the next enzyme, it is gamma glutamyl transpeptidase, that is GTP. Rise is very late and the peak value is between 7 to 10 days and this lasts longer, about for 4 weeks. This is helpful in the late diagnosis. Serum rise due to repair process source vascular endothelium from angioblastic proliferation. Now we'll see the other enzymes which is increased during myocardial infarction. We'll see acetylcholinesterase. There are two acetylcholinesterase. One is true acetylcholinesterase, another one is pseudocholinesterase. We'll see one by one. Coming on for acetylcholinesterase, this can act mainly on acetylcholine molecule. It is present in nerve endings and in RBCs. The normal serum range is 2 to 12 units per ml. The newly formed RBC will contain good quality of acetylcholinesterase, which is slowly reduced according to the age of the cell. Therefore, the acetylcholinesterase level in RBC will be proportional to the reticulocyte count. So coming on for the next type, that is pseudocholinesterase. It is a non-specific and can hydrolyze acyl esters. It is produced mainly by the liver cells. Normal serum level of pseudocholinesterase is 8 to 18 units per liter. The pseudocholinesterase level is seen in serum in reduced in viral hepatitis, cirrhosis, hepatocellular carcinoma, metastatic cancer of liver, and in malnutrition. Process of decreased plasma cholinesterase activity. This is during hepatic parenchymal disease, that is reduced synthesis, injection or absorption through the skin of such acetylcholinesterase as organophosphates, and during inherited abnormal cholinesterase variants with low biological activity. Causes of increased plasma cholinesterase activity is due to recovery from the liver damage and also due to nephrotic syndrome. Minor enzyme which is increased during myocardial infarction is histaminase. This enzyme histaminase occurs in different organs in various species. In man, however, normal plasma contains either very little amount of histaminase or none at all. But considerable amount of histaminase have been found in human heart muscles. Estimation of serum histaminase have been done by volumetric method. The normal value of histaminase is 0.12 to 0.76 pu per ml. We'll see the interpretation. Raised histaminase is found above 0.8 pu per ml in 97.14% of ECG proved cases of myocardial infarction. During myocardial infarction, the serum enzyme activity rises within 6 hours of myocardial infarction and persists for whole of the first week. By using these enzyme assays, we can find out the myocardial infarction. We'll see the next topic, that is enzymes as therapeutic agents. The medical application of enzymes is to treat enzyme-related disorders, to assist in metabolism, to assist in drug delivery, to diagnose and detect disease, and also during manufacture of medicines. So we'll see everything one by one. The enzyme used to treat disorders. This is the first one. The enzymes are used in 3 cases here. So coming on for the first one, it is to break the internal blood clots. And the second one is to dissolve the hardening of the walls of blood vessels. And third is to dissolve the wound swelling to promote healing. In some disorders like blood pressure or head or spinal injuries, there are chances of formation of the blood clots. These blood clots leads to the obstruction of the blood flow to the target organ. This can be life-threatening if it is in the brain or heart, which requires constant supply of oxygen and energy. The only way out then is to dissolve the clots. These clots are usually removed by the enzyme, by dissolving by the enzyme that can break them. So examples of such enzymes are streptokinase and urokinase. And similarly, when there is atherosclerosis, there is a hardening and thickening of the blood vessel walls. This can lead to heart problems if it is untreated. The best way is to decrease the fat intake and also dissolve the form thickenings. The enzyme such as ceratopeptidase can work well. For wound healing, the swelling formed might be painful and tend to form pus. The enzymes like trypsin, chymotrypsin and ceratopeptidase are used to dissolve the swelling. So then coming on for the next, it is enzymes used to assess metabolism. In old, the digestive capacity is low due to insufficient secretion of digestive enzyme. Hence, the digestive system cannot digest the food materials efficiently. In such cases, they can experience malnutrition, constipation and bloating, etc. To aid digestion, the enzymes like papain are administered orally after food for easier digestion. The next therapeutic agent is enzymes used to assess drug delivery. Some drugs need to penetrate deeper into the tissues for better action. For this, some enzymes are used along with the drugs in intramuscular injection forms to help the proper penetration of tissues. One such enzyme is hyaluronidase. This is a natural human enzyme present in human sperm. This can penetrate uterine tissue and fertilize with the ova. Here, the same enzyme is manufactured by rDNA technology and administered along with the drug to enable efficient drug delivery to the target site. Next is enzymes used to diagnose disorders. The enzymes which is present in the liver, kidney, skeletal muscle, heart, etc. leak into the blood during related disorders. So, by measuring the levels of the corresponding enzymes for their presence in high or low level in blood indicates a specific disorder. So, we can take an example, that is creatine kinase. This creatine kinase will be increased for muscle weakness and during injury. So, coming on for the last therapeutic agent, it is enzymes used in manufacture of medicines. There are immobilized enzymes, and this immobilized enzyme are used in the manufacture of many drugs and antibiotic. This is possible as enzyme convert the product molecules to drugs or starting material to drugs. All steroid drugs are manufactured by enzymes action on plant steroids. In today's topic, we have discussed about the enzymes which is increased during myocardial infarction and about the enzymes as therapeutic agents. Thank you.